Alright, hi guys. So welcome to this lesson, which is lead up from what we learned about heredity. So in the previous lesson, we actually learned about all the different terms. So we learned about things like alleles, we learned about things like chromosomes, genes, genotype, phenotype, homozygous, heterozygous, etc. So in this video itself, right, I'm actually going to focus on the concept on 16.2, which is exactly how are traits being passed down from our parents to ourselves. Now, for this part of the notes itself, right, you realize that there's a lot of drawing and a lot of concepts that you need to understand. There's really nothing much over here that you can memorize per se, but there's a lot of understanding to do. So without further ado, let's get started. So the lesson objective for today, right, is to construct what we call genetic diagrams. Okay? And also we need to learn how to predict the results of simple crosses with expected ratios. And last but not least, be able to explain why observed ratios of offsprings can be different from expected ratio. Alright, before we start, just want to ask you guys, what do you think a monk and a pea have in common? Fun fact, there's nothing in common. Okay, so basically I'm going to show you a video right now to help you explain about this thing called the Mendel's experiment.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett Square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase Y always overpowers his lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett Square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
Alright, as you can see from the video itself, Mendel, or Gregor Mendel, okay, who is a monk in the olden days, is actually the founding father of modern genetics. He was the one that actually first discovered about inheritance and used that through peas. Okay, so planting a lot of the plants, they can actually discover how certain genetic traits are actually passed down from generations to generations. So, monohybrid inheritance. Monohybrid inheritance refers to the inheritance of one characteristic based on two possible alleles. Now, let me just unpack the word a little. Mono refers to one. So when we say it's monohybrid, we're referring to just one particular thing. Hybrid, because there's a crossing. Crossing meaning to say that there's a mating process whereby there's a mixture of characteristics from both parents to the offspring. So it's a monohybrid inheritance. Inheritance meaning that you pass down certain traits to the offspring. Okay, so that's called monohybrid inheritance. Mono because you're only looking at one characteristic. Of course, there can be more characteristics that we look at it, but to make things simpler for our syllabus, right, we only look at one trait at one time. So what exactly do I mean by one trait at one time? For instance, we can be comparing about the height of the plant, or the color of the flowers, or the size of the leaves, etc. So you're only looking at one trait at each time. Remember, each characteristic is controlled by a single gene, but take note that each gene can consist of a pair of alleles, and some of the alleles over here could be recessive, and the other one could be dominant. Alright, so because of that, the behavior of the alleles, they can actually exhibit different what we call phenotypes. Now in Mendel's experiment, what he did was he actually found out, he actually compared a lot of various things. For the seeds of the plant itself, right, he actually went to compare about the form, okay, whether is it round or is it wrinkled. For the color, he also went to compare about it, the flower as well, the stem, the height of the stem, so on and so forth. So he actually found out certain very interesting ratio. So if you look at it, he actually managed to discover how certain behavior actually exhibits from generation to generation. What he did was he actually crossed purebred tall pea plants with purebred dwarf plants. Now, maybe let me just emphasize a bit on what exactly is the meaning of purebred. Basically, when we say that something is a purebred species, right, we're just saying that they are homozygous. Okay, so you learned in the previous lesson about homozygous and heterozygous. So by saying that it's pure, we're actually saying that it's actually homozygous to begin with. So that means that inside this pea plant, right, all the alleles in this tall pea plant will only consist of tall alleles, because it's a homozygous genotype. Okay, so likewise, the dwarf plants will only consist of dwarf alleles because it's a homozygous type, because it's pure. If it's not pure, we'll mention it in that question. So when he realized that when he crossed these two, right, the offspring that they obtained, okay, when he grew it out, it actually all became tall plants. Now that was one set of monohybrid inheritance, because he's looking at one trait, and the trait, the characteristics is actually the height. So this is what I call monohybrid inheritance. So we are looking at how the height is being passed down to offspring. And interestingly, all the offspring at that generation actually became tall. Now we call this the filial one, which is F1 generation. Now this is the first generation. Next, what he did was he allows the F1 generation to fertilize either through self-fertilization or not, and he obtained the seeds from it. So basically, he allowed the F1 offsprings to self-fertilize, and then he took out the seeds and he grew the seeds again. So basically, what we are looking at right now, right, is in this F1 generation, he's taking the tall species, allowing it to self-fertilize. So we are actually crossing between tall and tall. But this tall over here, just take note, they are no longer like the parents, because the parents were purebred. That means the parents were homozygous. But I'm not saying that the offspring here are homozygous. We don't know what they are. But I realized that when I cross them over here, the next generation of offspring that comes out, which I call the filial 2 generation, F2 generation, actually produce a ratio of about 3 tall plants to 1 dwarf plants, which means that out of all the plants that he grows, there's actually a very significant ratio of 3/4 of them are actually tall, only 1/4 of them are actually dwarf. So from this experiment, he actually found out a few things. Number one, he realized that there are certain characteristics that are dominant and certain that are recessive. So he actually found out that the tall trait is actually dominant. He didn't derive at this conclusion because tall is better than dwarf. Okay, that's not the purpose of saying whether something is dominant or recessive. He came to this conclusion because he realized that over generations and generations, it appears that the tall allele tends to be shown through the phenotype, because most of the plants will tend to be tall if the parents are actually tall. Okay, and even if you mix a tall plant with a short plant, a dwarf plant, the tendency for offspring to be tall is also quite significant. So that's when he realized that this trait itself must be quite dominant in nature. Now, do take note that during Mendel's time, right, he actually hasn't discovered about this thing called alleles. That's why he actually refers to them as factors. He's not too sure what exactly is the reason for this inheritance. He can deduce that there's something that is more dominant than the other one, and to him, he called them factors. Now, what he also derived is that these two factors, right, is actually being separated during gamete formation. For instance, if a parent were to pass down the genes to the offspring, right, it won't pass down the full sets of genes or the full sets of DNA. In fact, it only pass down one set of DNA. That's why we say that the two factors are being separated. And that means each gamete should only contain one factor. That's what we call the law of segregation. As a result, when the gametes fuse together during fertilization, it ensures that the zygote that's being formed contains of two factors. Now, this might seem a bit abstract, but if I were to break it into what you're more familiar with, which is alleles, basically what I'm trying to say is that if you take the example of a human, when there's a sperm as well as the ovum, we understand that inside the sperm and the ovum, there's only 23 chromosomes each. And but in a normal human, there's actually 46 chromosomes. So it's likely that these two gametes, right, only has half of what the normal cells would have. That's why you say that they only contain one factor by itself. Which means that inside this sperm, it will contain, for instance, one allele, for example, for height. Maybe just an example. Okay, for the human's case of height, it's a lot more complicated than this. So let's assume that he has one allele for height inside the nucleus. Likewise, for the ovum, there's only one allele for height. During fertilization, because it's a random process, we don't know which sperm will be able to fuse with the egg. So it's a random chance occurrence. Basically, once they fuse together, we know that when they form the zygote, within the zygote itself, now there will be two alleles that is representing for height. Okay, one that came from the dad, one that came from the mom. And that's what Mendel's experiment actually showed. But during his time, he referred to alleles as factors because he's not too sure about what exactly are genes, etc. So how do we show this entire process of, like what I've shown over here, how do we show that the sperm and the ovum actually can pass down the allele, and when they combine, and it actually results in the offspring? So what we do is we use something called a genetic diagram. Now a genetic diagram is used to explain how alleles are passed down to the offspring. And it also allows us to predict the traits that will be displayed by an offspring. So what I have over here is a very simple example of what it might appear. Now, for a purebred tall parent, like what I said just now, purebred means it's homozygous. That's why you see the genotype as big T and big T. For a short parent that is purebred as well, because purebred means it's homozygous, it will be small t and small t. According to what Mendel discovered, if the parents are of this genotype, they can only pass on one of the factors to the offspring. So when they form the gametes itself, right, only the gametes will only have one factor, which is one allele. So one of the gamete might have a big T over here, and the other gamete might have the other big T. So that's why at the start it was big T and big T. Whereas for the other case, one of the gamete might have a small t, the other gamete might have the other small t. And that's why when they fertilize together, when you just combine from a male and the female plant together, what you realize is all the offspring that is formed will be big T and small t. Okay, when you combine together, all the offspring that form will be big T and small t, which means that this is a heterozygous genotype because there's a combination of two different alleles. But in terms of the phenotype, because there's a big T over here, which is dominant, you realize that the phenotype that's being shown is actually tall. So all the plants here that's being formed will always 100% be tall because there's always this dominant allele that's found inside their genotype. Alright, so what I'm going to show you right now is what we are interested in our syllabus, which is how to use a genetic diagram to represent the entire inheritance that was being observed by Mendel. So just recap a bit about what Mendel observed. So basically for Mendel's experiment, he realized that if he were to cross two of the purebred parents together, he actually obtained all tall offspring in the first generation. But once he mixed all these tall offsprings, he realized that the next generation, which is the F2 generation, will have a combination of either tall or dwarf plants, and it's in a ratio of 3 tall to every 1 dwarf plant. So how can we represent this in what we call a genetic diagram? Now let me show you what exactly is crucial. In a genetic diagram, we need to be as clear as we can so that we can show how come we can derive this entire trait. So you notice in my notes, right, I have this set of tables on the left side. These are what I call headings. So let's start off first. In Mendel's experiment, what he did was he actually crossed a tall parent with a dwarf parent. Okay, so to represent the crossing or the mating process, we actually just put a cross in between them. So that's how we show that a tall and a dwarf parent actually cross together. Now the genotype of the parents, remember I mentioned at the very start, these are purebred parents. So if they are purebred parents, it means that they must be homozygous. So let's make some assumption. Assume that the allele for tall trait is represented by a big T, and the allele for dwarf trait, which is the recessive trait, is represented by small t. Why do we do that? So that we can represent the genotype. If you cannot decide what exactly is the alphabet that we are using, you can't really decide on the genotype. And we usually use a capital and a small letter to differentiate them. The parents of this entire system, right, they have a genotype of big T, because they are purebred, so they're homozygous. And the other parent has a small t, because it's a dwarf plant and it's purebred, so it's homozygous recessive. When they cross, we also put a cross over here. Now the next step is important, because if this is the genotype of the parents, right, when they form the gametes, we remember about the law of segregation, which means that they must be able to split up the alleles. So what happens over here with the arrows, this genotype is going to be divided into two possible types of gametes. We are talking about possible gametes. Okay, and for gametes, we always put a circle around it to represent it. It's like a cell by itself. The small t over here, likewise, it can only give rise to two possibilities, which is either small t or small t. Now, you realize that I will always show the entire splitting process, regardless of the fact that what I will end up with. Because for the tall plant, I always end up with big T in this case, but I still draw the two gametes. How do I know how the gametes will fuse? Because of random fertilization, the gametes will fuse randomly. You cannot decide which gamete should fuse with which one, because in reality, right, you cannot decide which sperm or which male gametes will fertilize which female gamete. So how does the randomization work? Basically, this particular gamete can possibly combine with this gamete, because one came from one parent, the other one came from the other parent. No matter what, you cannot say that both big T will combine back together, because both big T came from the same parent. So this is one possibility, and if they combine together, the genotype that we end up with is big T and small t. That's one possibility. Alternatively, some of you might say that, no, sure, but what if this one can combine with the other one? And the answer is yes, it's also possible, but in this case, it is still big T, small t as the genotype of the offspring. Now what about the other gametes? The other gametes of this big T can also randomly combine with this gamete to form big T and small t, and it can also randomly combine with this gamete to form a big T and small t. Now what I'm trying to show you over here is not to tell you that all the gametes are going to combine together. You cannot decide how the gametes will fertilize and fuse together. That's why we will have to come up with all the possibilities that could have happened. So this is how we derive the entire thing. To make this simpler for you guys, I would suggest that we label the gametes. Say this, let's call this label gamete 1, gamete 2, gamete 3, gamete 4. Just write rest in a small font so you can remember it. Basically, how will the random fertilization work? Basically, 1 will fuse with 3, 1 can also fuse with 4, 2 can fuse with 3, 2 can also fuse with 4. So this is what I call the 1 3 1 4 2 3 2 4 method. Okay, so you can just use the arrows to represent it. But of course, if you can remember how they should fuse together, perfectly fine. There's no need for you to remember it by this code. Okay, but if you need some help to remember, you can use this. So what exactly do you see over here? Now, all these 4 possibilities that can happen are all the same. All these will result in tall offsprings. But these tall offsprings are not the normal tall offspring, because they have the big T and small t genotype. Okay, so these are tall offspring as well. That's why in Mendel's experiment, he realized that all the offspring in the first generation, the F1 generation, are all tall. There won't be a dwarf plant in the F1 generation. Now, what happened next? Basically, in Mendel's experiment, right, what he did was he actually let the F1 generation self-fertilize. So likewise over here, we do need to represent what happens during the self-fertilization process. So I think in your notes there's a bit of typo, so I need to cancel away this entire tab on gametes, but I need to write something within it. So basically, now if you have to cross the F1 generation, what happens is that you have to take the tall one of the F1, you cross it, self-fertilization, so you cross it with a tall one. But what is interesting is that when these two things cross together, they actually have a different genotype from their parents. Because at the very start, I told you that the parents that we have over here, one of it is actually tall also, but it's big T and big T. But that's because it is purebred. But now we realize that all these tall phenotype, right, actually belongs to a heterozygous genotype. This means that we are looking at a combination of big T, small t crossing with big T, small t. Now with this combination, that's when we can derive the gametes. So I just want to shift your gametes to a lower column over here. Now what exactly happens here? When the gametes are formed, we are looking at the formation of a big T or a small t from this combination. We are looking at a combination of a big T or a small t from this combination. What does this mean? If you recall about my random fertilization, if I try to let the F1 generation fertilize itself, I cannot decide which gamete will fuse with which one. I can decide that it's going to be 1, 3, 1, 4, 2, 3, 2, 4. So 1, 3, 1, 4, 2, 3, 2, 4. And there you have it. I realized that I actually have another combinations of genotypes. And what's interesting now is that not all the genotype represents tall plants. The first one, yes, is tall. The second one, yes, is still tall. The third one, yes, is still tall. But the last one is actually a dwarf phenotype because it only comprises of recessive alleles. What does this mean? This means that out of all my plants, if I look at the ratio of the F2 generation between the tall and the dwarf plant, I will see a very distinct ratio of 3:1. That means for out of 100 plants that I have, 75 of them is likely to be tall, but 25 of them is likely to be dwarf. That's the power of genetic diagram. It allows you to know how the entire crossing should happen. And this is the entire working that you should be able to show. Now, of course, there's an easier method to do this entire genetic diagram. It's what I call the Punnett Square. Now, the Punnett Square works in 9 grid boxes. So what it does is it do a very fantastic job in explaining how genetic crosses come about. So for the first cross that was mentioned just now, if we were to cross between a tall with a dwarf parent, assuming that both are pure, how to show what could be the offspring's genotype? We can just draw a grid of 9 by 9 without using this particular grid. What I can do is that if I know that the tall plant is actually a purebred plant, I'll know that it actually has a genotype of big T, big T. If the dwarf plant that is purebred, it should have a genotype of small t, small t. Now how do I represent it in the Punnett Square? Basically, looking at the top column, the top row over here, we can use this to house the 2 gametes that can form from either one of the genotype. For instance, let's say that there's a big T here, and there's a big T possibility of a gamete being formed. This is very similar to like in the previous page, right? We said that the genotype differs with 2 possible gametes. Small t over here, small t over here, based on the other parent. Now, how will the cross look like? Basically, just combine the different alleles. This will give you a genotype of big T and small t. This will give you a genotype of big T, small t, big T, small t, big T, small t. And therefore, I can realize that all offsprings will have the big T, small t genotype and will be tall. Right, so this is one way that I can realize the entire thing. Do take note of how I represent the gametes and how I try to differentiate them from the genotype. Gametes wise are always in a circle. So we always do this to represent a gamete. But if you remember, Mendel also did another experiment. He actually allowed all these offspring over here to cross with themselves. So right now what we have again is tall crossing with tall again. Now what we show you in the genetic diagram. But this time all the tall over here, they're actually big T, small t. They're actually big T, small t. Why? Because they came from this generation. So since all the generation over here is big T, small t, when we cross over here, it should also be a big T, small t with a big T, small t. Now how do I explain what will be the offsprings? Same thing, we draw a Punnett Square. If you notice by now, there's no need for the top left hand column, so I'm just going to miss it out. And how do I represent it? Basically, from this big T, small t combination, I can have a big T allele and a small t allele. From this one, I can also have a big T allele and a small t allele. When they fuse together, I can end up with big T, big T, small t, big T, small t. Or if I look at this combination, I can end up with a small t, small t. So through this thing, likewise, like what I did just now in the genetic diagram, I can also tell that the tall plant in ratio to the dwarf plant is a ratio of 3:1. So that's what we can use the Punnett Square to help you with. Right? So these are just 2 different methods that we can use to derive how traits are being passed down. Of course, there needs a lot more self-practice. That's why what we are going to do now, we're actually going to embark on another self-practice on the next page of the notes. Do take note of what the question actually wants you to show, so that you know whether you are drawing a genetic diagram or are you drawing a Punnett square. Because as you can tell, it's a lot of work to draw a genetic diagram, but for the Punnett square, it's a lot more straightforward. Let's take a look at our first example. In mice, the allele for long fur, capital L, is dominant, and the allele for short fur, small letter l, is recessive. Now, you are actually required to construct a genetic diagram to show the ratio and genotypes of the F1 generation when a homozygous long fur mouse is mated with a homozygous short fur mouse. Before we start off, let us do some working. Capital L is dominant, small letter l is recessive. If the first mouse over here is homozygous long fur, homozygous will tell you that it is actually made out of 2 of the similar allele, and since it's long fur, it will be 2 capital L. Likewise, homozygous with short fur will be 2 small l, and that will be the genotype of the mouse. Not the alleles, but the genotype. You're supposed to construct a genetic diagram, so not a Punnett square for this question. And you're supposed to show the ratio and genotypes of F1 generation. Okay, so you're going to do one set to show the F1 generation. Next, a male and female from the F1 generation is then mated to form the F2 generation. Likewise, you are required to construct a genetic diagram to show the ratio and genotypes, but now of the F2 generation. So these are 2 important things they need to do for this question. Now, there's a need for us to be able to express our working in a neat and orderly manner. That's why I will encourage you to try as much as possible to take the effort to make sure your working is neat and tidy. First, to show that the parents are actually crossing with one another or mating with one another, we actually have to write down this first. Parents phenotype. Because we need to set the context first to know how are the parents actually related. So for the parents phenotype, one of them is actually long fur, the other one is actually short fur. Alright, so we start off with this context based on the question. Now we are able to derive the genotype because the question actually mentioned that what exactly is the genotype, because it mentioned about homozygous. So I know that the parents genotype is as follows. Capital L and small letter L. Right, so that's the parents genotype. Now the parents must be able to give rise to gametes. So for the gametes that they can form, right, I should also know that if it's capital L and capital L genotype, they can only form either capital L or the other way is still capital L, but we still have to express it out because to show that we understand that it can be separated in this manner. For this one, it will be small l and small l. Now for the offspring to be formed, there must be some fertilization, and this is what I call random fertilization. Now in random fertilization, what we mean is that the gametes are now going to combine, but we are not too sure how will they behave like. So gamete 1 can combine with gamete 3 to form capital L, small letter l. There's no need to put a circle anymore because now you are showing a genotype. Gamete 1 can also combine with gamete 4 to form capital L and small letter l as a genotype. Likewise, and likewise. What do you end up with over here in this entire row is we actually end up with the F1 generation's genotypes. Because we are trying to see what is the F1 generation genotype, and it could be, in fact, it's all the same genotype, which is all capital L and small letter l, which means that they are all heterozygous. The question is asking you to show the ratio as well as the genotype of the F1 generation. So up to this point, we're actually showing the ratio, the genotype of the F1 generation. How do we show the ratio? The ratio of F1 genotype, since all of them are capital L and small letter l, will be all big L, small l. So all of them will actually be what I call heterozygous genotype. Okay, so that's how we do for the first set of questions. Okay, if you need some more time to digest this, I suggest you pause the video, look through it again before you move on to question B. For question B, what we are doing now is actually we are trying to cross the offsprings from the F1 generation, taking a male and female mouse and letting them breed. Now this time round, we are also expected to draw a genetic diagram to show the ratio and genotypes of the F2 generation. Basically, this time round, the parents, which is actually the F1 generation now, also has a certain phenotype, and is that they are all long fur because they are all of the heterozygous genotype. So if this is their phenotype, we also need to understand what are their genotypes. Now, as I've mentioned before, they are of the heterozygous genotype, so they should be a capital L with a small letter l, crossing with a capital L and a small letter l. What's next is to form the gametes, because from here you can derive what are the gametes. It's possible to get a capital L or small letter l gamete. It's possible to get a capital L gamete or small letter l gamete. These are what I call the gametes. Next is when fertilization happens. In fertilization, the gametes will randomly combine with one another. But we don't have a scenario whereby it's this and this, because you can't have similar gametes from the same parent fertilizing each other. Right? So it doesn't make sense. It's like two sperms cannot combine together, right? So in this case, my 1, 3, 1, 4, 2, 3, 2, 4. You notice that if I try to draw it neatly, right, it will make my entire work a lot easier to understand, and that means that in the exam, right, when I check my work, it's a lot easier to understand also. This will result in capital L, this will result in capital L, capital L, small l, and small l, small l. These are what I call the F2 genotype, because it shows what are the genotypes of the offspring. From this, I can derive about the ratio, because the question is asking for the ratio and the genotype of the F2 generation. The ratio of F2 generation, do take note, we are comparing the genotype. So we are looking at the types of genotypes there are. And it's actually a capital L combination, or a capital L, or a small l. And you will realize that from this set of question that there's actually two chances to get the big L and small l. That's why the ratio is actually 1:2:1. But take note, this is the ratio of the genotype. Why do I say that? Because if you are comparing the ratio of the phenotype, so this is extra work, ratio of phenotype, you're actually expected to understand that this combination as well as this combination will both give you the long fur. So if I were to compare phenotype, which is long, compared to short fur, it's actually a ratio of 3:1. But in this question, we're actually only focusing on the genotype itself. So there's no need for you to explain to me about phenotype. If not, the next two questions are for you to try out and also to attempt the assignment that's been put up online. Okay, you do have to take a photo of this piece of work on the next few pages and try out for yourself. But of course, take note if the question is asking for genetic diagram or Punnett Square. That's all for today. See you.